Ophthalmology meeting in New Orleans, Louisiana. It's great to be back and see so many doctors and colleagues here connecting once again in person and getting the feel for once again being on an exhibition. I'm also delighted to welcome live today Dr. Mitchell Brigel. Dr. Brigel was supposed to be here in person, but a last minute scheduling change had him away, and so he's joining us today from a undisclosed location. So, welcome, Dr. Brigel, who many of you may know. He's been extremely involved in the pharmaceutical industry for many, many years in translational medicine and helping bring life saving and sight saving therapeutics to market over the years. And also, he is an officer of the ICEV organization, the Clinical Electrophysiology of Vision organization, and so it's delighted to welcome Dr. Brigel here this morning. And he's also done some research with the Redival device, particularly in diabetic retinopathy and macular edema. So, Dr. Brigel, welcome.
Thanks, Jim. Sorry I couldn't be there in person. Would have been nice to see you and colleagues in New Orleans, but hopefully
Absolutely.
next year.
So, Dr. Brigel, can you tell us a little more about your work in the area of diabetic retinopathy?
Yeah, well, I've been trying to develop drugs, new treatments for diabetic retinopathy, but specifically with regards to the Redival, I read with great interest the paper that April Ma published on diabetic retinopathy and using Redival as a diagnostic and showing that there was a very good relation between the Redival DR score and DRSS severity, diabetic retinopathy severity. And in conversations with Quentin Davis and you and Dr. Ma, we decided to explore that further. Specifically, if you look at the plots of DRSS versus the Redival DR risk score, while there's a correlation, there's variability. So for any DRSS score, some patients have very good-looking ERGs and others have more affected ERGs. And the question we posed is whether that's just noise in the measurements or whether the response, the physiologic response that you get from the Redival device actually adds prognostic information to what you can see when you look at the fundus. And so what we did was take 250 patients that participated in that initial study from the Atlanta VA, and we did a retrospective chart review over 3 years to see the impact of baseline DRSS score and Redival score on subsequent need for intervention for diabetic retinopathy. And the results were published in TVST in 2020 and were quite striking. The Redival risk score actually was more predictive of the need for intervention for diabetic retinopathy over the subsequent 3 years than was the DRSS score per se. But to me, even more interesting was that the combination of the two was very powerful. And to just put that into perspective, if you have a anatomic DR with visual risk, so either DME or DRSS 53 or above, and your Redival score is negative, your risk of actually developing the need for intervention is as low as if your DRSS score was mild or moderate. Whereas if both were positive, there was like an 80% chance that over the next 3 years you would have needed intervention. So we published a lot of results, but the bottom line is that adding the functional information you get from the RED-EVAL to the DRSS score really helps you manage your patients. You can better see who's at high risk for progression and who needs more frequent visits and perhaps intervention, using both those than just using the DRSS score alone. So I think it's very important both scientifically and clinically in managing patients.
All right, thank you. You know, I think with the ophthalmologists and optometrists who are listening today probably harken back to their medical training when they think about the visual electrophysiology and they see these huge desk-sized pieces of equipment that require dilation and electrodes being placed directly on the eye, and you know, a specialized technician able to do the work, and maybe even a special room with a Faraday cage kind of arrangement around it to keep the noise down, and then kind of difficulty. Right. How do you interpret all these squiggly waveforms, and how has the RED-EVAL changed that from your experience in these?
Yeah, I mean it's groundbreaking. I mean, we did a subsequent study for a drug that we were developing for diabetic retinopathy, a multi-center randomized clinical trial where we put this device in 20 institutions, that were participating in the trial, none of which had the RED-EVAL before. They took, a maybe 1 hour training course online, got certified that they could do it correctly, and we got beautiful data from all those sites. The device, especially for DR, the Flicker ERG does not require dark adaptation, and as you said, it uses skin electrodes. So it's revolutionized the use of the ERG. The footprint of the device is minuscule. It could be placed at a desk next to your laptop. It's really taken the ERG to places that it wouldn't go before, both in terms of clinical trials and in terms of clinical practice. There's no need for a lot of training. If you're using it for predicting DR, the score comes out automatically. So I think it's been a huge boon for the use of ERG, beyond the traditional use in screening for inherited retinal diseases, and brings it into more common diseases like diabetic retinopathy, and should really expand the practical use of ERGs in conjunction with imaging. Yeah.
And the use of the skin electrodes that just attach to the skin very quickly and easily. It said you can test the patient in less than 5 minutes, both eyes, without dilation. It's just very easy and very comfortable for the patient. So that's in April Ma's study that you referenced. It had a success rate of over 99% of the patients tested. So it's also very useful for patients who may have opacities of media and other things that would make very, very small pupils of 2 millimeters or less really not an impediment to getting a result with this device. So, how did you? What do you think that ophthalmologists and optometrists who are managing and seeing patients with diabetic retinopathy, how would you see this impacting their care? How would they actually use this in a practical manner?
Yeah, I mean, I think, as we all know, grading DRSS is not an easy task. I mean, you can look in the eye and definitely see background retinopathy. And for ophthalmologists and optometrists, how do you manage that? Can you see the patient once a year? Do they need more frequent visits are they about to fall off the cliff and lose vision either to DME or to diabetic retinopathy? That's what you want to do to help your patient. And having a RED-EVAL evaluation allows you to take someone with background retinopathy who's seeing just fine and say, this patient is at risk to lose vision over the next year or two, and therefore I need to see them more frequently. Or this patient, has a nice ERG, and I can see them once or twice a year and not put the patient at risk. So it really helps you manage the patient in a way that's beneficial to the patient, and eventually to the healthcare system, and hopefully that they never lose vision to their diabetes.
That's awesome. Thank you, and thank you very much, Dr. Miguel. It's also interesting to note that we're talking about the use of diabetic retinopathy, but this really is a handheld, full function like ERG, flash VEP system that does all the standard ICEV tests, both scotopic and photopic, as well as photopic negatives for ganglion cell measures and so forth. And so perhaps you can comment a bit on its use in those areas, both in pediatrics all the way up through geriatrics.
Yeah, well, I mean, the ERG has utility in monitoring the physiology of the retina that's been validated since post World War II. I mean, it has use in vascular diseases. I mean, Mary Johnson published work back in the 80s showing, its use in retinal vein occlusions and diabetes we've talked about. Certainly now in looking at ganglion cell function in glaucoma, we have measures that are able to look at the health of the different layers of the retina in a quantifiable manner that probably precedes structural damage. So, I think we're used to in glaucoma looking at visual fields and looking at the optic disc. Well, now we have OCT looking at the nerve fiber layer, and we have the photopic negative response to look at the health of the ganglion cells. And the visual fields, we all know, are quite variable and difficult for the patient. The photopic negative response can give you an indication of the health of the ganglion cells and the nerve fiber layer with much less variability and with much less effort on the part of the patient. With a quick ERG, you can monitor, the physiology of the retina in addition to looking at the structure of the nerve fiber layer. So again, just like for diabetic retinopathy, I think the two together are of much more use in managing a patient than just looking at an OCT or looking at the disc.
Thank you. One final question is about the study that you did, particularly the drug study that you were involved with. You used the different modalities of testing the patients, including RED-EVAL, as well as OCTA and so forth. Can you comment about the prediction of progression of those other modalities as compared to DR score RED-EVAL?
Yeah, that we haven't published that yet. We presented at ARVO in 2020, and we really should write it up. We in this clinical trial, randomized controlled clinical trial of 150 patients, we measured OCT angiography, we measured ultra wide field photography, looking at the far periphery, and we measured the RED-EVAL DR. And over the course of the year of the trial, 40 patients had progressed to either proliferative diabetic retinopathy or to DME. At entry, they had DRSS scores between 43 and 53, so they had moderate to severe diabetic retinopathy and no DME. And we found that the Redival was, again, more predictive than either of the structural measures in who is going to progress. And again, like we found in the VA study, but in this time in a prospective manner, we found that the combination of non-perfusion score on OCTA combined with Redival was, again, very predictive of subsequent progression, such that if both were negative, there was only a 3% chance of progression, whereas if both were positive, there was over a 70% chance of progression over 1 year. So again, showing the power not only of electroretinography in prognosis, but the combined structure function as having really helping managing patients.
And one final, just general question, Dr. Bigel. You know, if you're an ophthalmologist or an optometrist and you're reluctant or reticent about perhaps introducing the Redival technology into your practice, what would you have to say to them to help with that perception?
Yeah, well, I mean, you know, the proof's in the pudding. How does it help? Does it help you manage your patients better? And I think there's more and more evidence coming out that adding functional measurements from the Redival to what you can see when you look in the fundus or to new imaging techniques helps you manage your patients better. And it's, as Jim has said, it's the testing now is not burdensome on the patient. It doesn't take a lot of room in your clinic space, and it's quick to do. And so I would encourage that you can manage your patients better if you have this device as part of your clinical service.
Okay. Well, I thank you. I appreciate your time today. As you can see, we're here at the American Academy of Ophthalmology in New Orleans, Louisiana. You can see the hustle and bustle behind me of people here at the show, all masked, all vaccinated, and all as safe as we can be. Hopefully it's harbinger of things to come as we over time reopen totally to the way things were before the pandemic. So I thank you, Dr. Bigel, for your time this morning. I thank you more for your perception and for all the work that you do to help patients preserve their vision. Thank you very, very much.
Thanks, Jim. My pleasure.
Okay.